Hello, grade 12 students. So last time pinag-usapan natin yung transcription. Paano ba gumagawa ng messenger RNA ang ating nucleus? But today, we will have a different topic, which is translation. So in this stage, the mRNA is decoded to build a protein or a chunk or a subunit of a protein that contains a specific series of amino acids. Okay. In an mRNA, the instructions for building of polypeptides or RNA nucleotides read in groups of three. These groups of three are all called codons. So here we have three nucleotides. So this is one codon. We have another three nucleotides here. This is the second codon. We have the third codon here. And the list goes on and on. Okay? Basta ang tatandaan, kapag sinabi nating codon, sila ay mga nucleotides na nakagrupo sa tatlo. Okay? And itong mga codons na to, they are instructions to create amino acids. So for example, AUG means methionine, yung amino acid na makukuha natin sa kanya. ACG, threonine. GAG, glutamate. CUU, leucine. CGG, arginine. AGC, serine. And UAG for the stop. Okay? So stop is not amino acid. Ibig sabihin natin dyan na natigil yung pag-synthesize ng amino acid. Stop. So tumigil yung pag-synthesize ng amino acid. Now, there are 61 codons for amino acids, and each of them is read to specify a certain amino acid out of the 20 commonly found in proteins. One codon, AUG, specifies the amino acid methionine. Okay, so we have methionine here, AUG. So, the amino acid methionine also acts as the start codon to signal the start of the protein construction. Okay? So, kung wala si AUG or kung wala ang methionine codon, ibig sabihin hindi makakaproceed sa pag-start ng amino acid synthesis. Kasi AUG is important in signaling the start of the protein synthesis. Now, there are three more codons that do not specify amino acids. These are the stop codons. So specifically, we have the UAA, UAG, and UGA. Ayan po. So stop codons sila. They tell the cell when a polypeptide is complete altogether. So altogether, this collection of codon amino acid relationships is called the genetic code because it lets cells decode an mRNA into a chain of amino acids. And a chain of amino acids is a peptide. Okay? A long chain of amino acids is a peptide. Now, how is an mRNA read to make a polypeptide? Okay? Paano ba binabasa ang mRNA para makagawa tayo ng isang polypeptide or ng isang protein? Okay, now there are two types of molecules with key roles in translation. These are your tRNAs and the ribosomes. So transfer RNAs or tRNAs are molecular bridges that connect mRNA codons to the amino acids that they encode. Okay, one end of each tRNA has a sequence of 3 nucleotides, so called an anticodon, which can bind to a specific mRNA codons. Alright. So, ito po. Yung isang end ng tRNA ay tinatawag natin na anticodon. Okay po. And the anticodon complements a specific codon. Okay? So, for example, ano yung complementary ng AAG? Okay? So, by using Chargaff's law, so we have UUC. Okay, makikita natin dito na meron tayong UUC dito sa mRNA. So, This tRNA corresponds to this part. Okay po. So ayan. So kakabit yung tRNA dito and meron siyang daladalang amino acid sa taas. Okay. Ayan po. Now, ribosomes are the structure where polypeptides or proteins are built. They are made up of protein and the ribosomal RNA or the rRNA. Each ribosome has two subunits, a large one and a small one, which come together around an mRNA. Kind of like the two halves of a hamburger. And yung palaman niya ay yung mRNA. So think of this as a hamburger bun. And dito, sa gitna, ang palaman niya ay ang messenger RNA. Okay. Ayan. So the ribosome provides a set of handy slots where tRNAs can find their matching codons on the mRNA template and deliver their amino acids. And these slots are called the A, P, and E sites. Not only that, but the ribosome also acts as an enzyme, catalyzing the chemical reaction that links amino acids together to make a chain. Okay po? So yan. So meron tayong tinatawag na EPA slots. Okay? So nandito yung E. Okay, nandito yung E slot, nandito yung P slot, and nandito yung A slot. Okay, now these slots are specific. Okay po. So, dyan kumakabit yung mga transfer RNA. Okay, now let's talk about the formation of translation initiation complex. So, nasa initiation stage na tayo. So, initiation, the ribosome assembles around the messenger RNA to be read. And the first transfer RNA carrying the amino acid methionine, which matches the start codon, AUG, ayan, kakabit siya. Okay? So, nandito yung ating start codon. Meron tayong anti-codon na complementary dun sa ating codon. So, we have AUG here. The complementary is UAC. And the anti-codon also carries the methionine. Okay? Ayan. So, that is the initiation complex. So, this setup is called the initiation complex. So, it is needed in order for the translation to get started and occur. Okay? Ayan. So, ganyan po. Now, let's talk about the extending or extension of the chain, which is the elongation process. So, in elongation is the stage wherein the amino acid chain gets longer. So, in elongation, the mRNA is read one codon at a time, and the amino acid matching each codon is added to the growing protein chain. Ayan. Each time a new codon is exposed, a matching tRNA binds to the codon, and the existing amino acid chain is linked onto the amino acid of the tRNA via a chemical reaction. And the mRNA is shifted one codon over in the ribosome, exposing new codon for reading. Ayan. So, tingnan nyo dito sa ating GIF. So, di ba, nag-start tayo with methionine. Ngayon, yung methionine, ayan, dudugtungan siya ng valine gamit yung peptide bonds. So, kapag nakadugtong na yung peptide bond, yung codon, yung anti-codon, rather, matatanggal na siya dito sa P site. Okay? And uusog naman yung nasa A site papunta sa P site. Okay po. Then, mababakante yung part na to. Okay, kapag nabakante yung part na to, that is the time para pumasok naman yung panibagong transfer RNA. Okay, kasi exposed na yung side na to. kung saan merong codon. So, ganun po. So, ganun ang process ng elongation. Hanggat meron pa tayong codon na nag-correspond sa isang amino acid, tuloy-tuloy lang po ang elongation. Okay? Yan. During elongation, tRNAs move through the A, P, and E sites of the ribosome, as shown here in the picture. So this process repeats many times as new codons are read and new amino acids are added to the chain. So remember, dito tayo sa P site. Ayan, dito yung part kung saan na idagdag yung methionine. Okay. Now, yung E site natin is exposed. Since exposed yung E site natin, may magbaband dyan na transfer RNA. Okay? So, expose yung codon dito sa A site, papasok yung corresponding na transfer RNA para supplyan ng anticodon. Okay? So, ayan. Nandito na. Perfect fit. So, itong daladalang amino acid, gagawa ng chemical bond dito sa taas. Okay? So remember, the ribosome helps in catalyzing the production of chemical bonds for the amino acids. So ayan. So magkakabit yung amino acids na to. Okay. Eventually, itong amino acid or itong transfer RNA rather, dito sa P site, ayan, wala na siyang daladalang amino acid. So, kailangan na niyang kumalas. So, mag-move siya pupunta dito sa E site. Okay. And sa E site, kapag nandito na siya sa E site, kakalas na siya. Okay? So, matatanggal na. Ayan. So, it's just a continuous process. So, patuloy lang yung pag-elongate hanggat meron pang mga codon na nag-mimin na merong amino acid dapat. Okay? Now, ang sunod na part after elongation is the termination. Now, paano natatapos yung ating translation? So, syempre, meron tayong tinatawag na termination. Okay? Termination is the stage in which the finished polypeptide chain is released. It begins when a stop codon, either of the 3 stop codons that we have, remember, UAG, UAA, or UGA. So, these are stop codons. So, when a stop codon enters a ribosome, it triggers a series of events that separates the chain from its tRNA and allow it to drift out of the ribosomes. Okay? So after termination, the polypeptide may still need to fold into the right 3D shape. So under the processing, such as removal of amino acids, so it gets shipped to the right place in the cell. So, or combined with other polypeptides before it can do its job as a functional protein. Okay po? So here we have the stop codon. Okay. Then we have the release factor. Now, the release factor is a type of transfer RNA that doesn't have any amino acids attached at its head. Okay? So, wala pong amino acid. Kung mapapansin nyo dito. Unlike the other transfer RNA, merong corresponding na amino acid. Okay? Ang stop codon, pag stop codon na, meron nang kumakabit na release factor. The release factor will signal the ribosome that the process of translation is already finished. Okay? So, and that's it. That ends the whole process of translation. Okay? Ibig sabihin, tapos na. Nakagawa na tayo ng isang peptide chain. Okay? And ngayon, yung chains natin na to, meron na tayong peptide. So, depende kung magiging primary protein siya, secondary protein siya, tertiary protein or quaternary protein para maging functional siya. So, yung mga peptides na to, naggugrupo-grupo pa sila. Okay po. Pero hindi na yun sakop ng ating discussion today. Ang pinakita lang natin ngayon ay kung paano nagkakaroon ng translation mula sa isang simpleng messenger RNA na merong daladalang mensahe hanggang sa makagawa tayo ng isang produkto na tinatawag natin na protein. Okay? So, ganun ang nangyayari. Ayan. Now, to give you a summary of our discussion, so remember transcription, the job of transcription is to create the precursor mRNA. Now, the precursor mRNA are not yet suitable to go outside the nucleus. Thus, it undergoes splicing, 5' copying, and the addition of poly-A tail para maging mature mRNA. And ang mature mRNA, ang lahat ng codes niya ay exons na lang. So, since exons lang yung nandun, yung mga introns wala na siya. So, remember the differences of exons and introns? Exons are the coding. Ibig sabihin, importante sila para sa gene expression. Since dala-dala nila yung codes para sa specific gene. Yung non-coding naman, sila yung mga introns. Okay? Yung mga introns, wala silang dala-dalang meaningful na instruction. So, kaya nga sila tinawag na non-coding. Okay? Now, kapag ayos na yung tatlong processes, yung splicing, 5' copying, ayan, and the addition of poly-A tail, pwede nang lumabas si mature mRNA. So, tandaan natin na ang mRNA, ginagawa siya sa nucleus. So, lalabas ito. Lalabas ang mRNA papunta sa cytoplasm. Kasi, matatagpuan natin ang ribosome sa cytoplasm. Diba? So, ayan po. So, yung mRNA, pupunta siya kay ribosome. Okay. Ang ribosome, meron tayong dalawang units. Okay, the small unit and the large unit. So, ayan. Makikita nyo naman. Para makapag-start tayo ng translation, kailangan natin ng codon for methionine. And after that, dahil sa process of initiation, elongation process will proceed. Ibig sabihin, hanggat meron pang anti-codon or meron pang codon dyan na merong corresponding amino acid, magpuproceed lang yung elongation natin. Okay? Magtatapos ang translation kapag naabot na nung ribosome yung tinatawag natin na stop codon. Okay, kapag naabot na yung stop codon, maglalagay ng anti-codon na release factor. Okay po? Yung release factor natin, wala na siyang daladalang amino acid. Ang ginagawa niya lang is to signal the termination process. So, kakalas na yung polypeptide na nagawa natin. Okay? Or the peptide na nagawa nung translation. So, i-release na to. Ayan. And that's it. That ends our discussion about translation. Very brief and concise. Diba? So, if you have other questions or concerns, just simply message me sa aking Facebook or pwede naman kayong mag-email. Okay, so salamat my dear grade 12 students sa pakikinig. And that ends the recorded discussion. So, thank you and goodbye. Okay, good luck sa exams. Sana makapasa kayo lahat. Or better, sana makataas kayong lahat. So, yun lamang po. Goodbye na.